Okay, Assalamualaikum and good morning everyone. So today we will learn about microbial growth response to food environment, okay? So today we will recall back what we have learned during your class, your microbiology class, okay? So microbial growth usually refer to the increase of cell number and not in cell size. Okay. So basically, when we talk about microbes, food poisoning, we usually talk about the increase of cell number. Okay. The most bacteria you have in the food, the severe the case of food apa nama ni food poisoning. Okay, bacteria grows and divide by binary fission, a rapid and relatively simple process. So we know that when we have the graph of the growth curve of the bacteria, we have log phase, stationary phase, and death phase. Okay, so we will go one by one. We will see one by one for the growth characteristics. Okay, so bacterial division occurs mainly by binary fission, just like I said previously in previous slide. A few reproduced by budding, usually yeast lah basically, and then the generation time required for cell to divide and its population to double. So generation time varies considerably. E. coli divides every 20 minutes. Most bacteria divides to 1 to 3 hours. Some require 24 hours to divide. So it depends on the bacteria species, okay? So microbial growth, which is kita tengok phases of growth. Bacterial growth curve, when bacteria are inoculated into liquid growth medium, we can plot of the number of cell in the population over time. So we go into lag phase, which is the period of adjustment to new condition. Little or no cell division occur, population size does not increase. That's why dia straight dia tak ada apa-apa peningkatan kat situ. Phase is intense of metabolic activity in which individual organism grow in size and it may last an hour or several days. Okay, so we can see here is the So we can see here is the lag phase. Okay, lag phase kat sini. Tak ada apa-apa yang berlaku kecuali dia macam tadilah. And then the binary fission, kita tahu bahawa cell elongates and DNA will replicate here. Kita ada cell wall, ada plasma membrane, DNA, and then dia will start replicate, and then cell wall and plasma and membrane begin to divide. So kita akan nampak kat sini, and then cross walls form completely around the divided DNA. And then cell will be separated. Okay, so kalau ikut kat sini, this is diagram of sequence of cell division, and this is a thin section of a cell of Bacillus linkiniformis starting to divide. We boleh nampak ada partially formed cross wall kat sini. Okay, and then we can see here the DNA nuclear DNA here and the cell wall. Okay. So growth of bacteria culture usually in logarithmic representation. We can express the number of cells in bacterial generation as 2n. N ni yang mana dia punya bakteria lah. Where n is the number of doubling that have occurred. So for example here, arithmetic number of cells, we have 1 here, 2 to the power of 0. So kita akan nampak bakteria tu ada 1. When 2 to the power of 1, bacteria ada 2. And then dia double lagi 4. And then dia double lagi dapat 8. Okay, and so on and so forth. Okay, so that is like phase. and then bacteria adapt themselves to growth condition. It is the period where the individual bacteria are maturing and not yet able to divide. During the lag phase of the bacteria growth cycle, synthesis of RNA, enzymes, and other molecules occur. So in this phase, the microorganisms are not dormant. So this sedang synthesize ada RNA, enzyme, and other molecules occur. So the log phase, kita tengok log phase pula. Log phase begin to divide and generation time reaches the constant minimum period of most rapid rapid growth. So yang ni number of cell produced more than number of cell dying. So cells are at high metabolic rate, metabolic activity, cells are most susceptible to adverse environment factor at this stage, which is for example radiation or antibiotic. Okay, so kita tengok pula dekat stationary phase, which is the straight phase here. Straight dekat sini juga, sama macam lag phase. What happened here basically, number of cell produced equals to number of cells dying. Sebab tu dia straight. Okay, overall cell number does not increase, cell division begins to slow down. Factors that slow down microbial growth, which is accumulation of toxic waste, sebab dia dah lama dia dah grow dalam tu, dia dah produce toxic materials, maybe the acidic of pH of the media, and then limited nutrients sebab dia dah guna nutrient dalam media tu, insufficient of oxygen supply because they have used the oxygen to produce the cells previously. Okay, and that's which is decline phase, number of cell dying more than more than number of cell produced, which is the population size begins to decrease. Cell number decrease at logarithmic rate, cell lose their ability to divide, and a few cells may remain alive for a long period of time. Okay, now we go to the growth requirement factors, which is intrinsic factor and extrinsic factor. Intrinsic is the inside factor, extrinsic is outside factors. Okay, for intrinsic factors, which is the pH, moisture content, oxidation, reduction potential, antimicrobial constituents, and biological structures. So this is the inside factors. Okay, inside factors. Okay, pH, effect of pH will affect enzyme and nutrients and other environmental factors like temperature, salts, and agar. No non-pathogen grows below pH of 4.6. For example, Clostridium botulinum. And then most bacteria prefer neutral pH 6.5 to 7.5. Moles and yeast grow in wider pH range but prefer pH between 5 and 6. Acidity inhibit usually most of bacterial growth and is frequently used for food preservation. For example, pickling kat sini. Alkalinity inhibit microbial growth but not commonly used for food preservation. Acidic product of bacteria metabolism interfere with growth. Buffer can be used to stabilize pH. And also pH organism can be classified as acidophiles, which is acid-loving. Bacteria grow at low pH 0.1 to 4.5. Which, for example, Lactobacillus produces lactic acid, tolerates mild acidity. Neutrophils grow at pH 4.5 to 8.5, include most of human pathogen. Okay, why do you think they prefer the pH like this? Okay, something to think about. Nanti boleh fikir. So this include most human pathogen, alkaliphiles, alkali-loving, grow at alkaline or high pH 7 to 12 or higher. For example, Vibrio cholera and Acinetobacter faecalis, optimal pH is 9, and Solid-Bacterium agrobacterium grow at pH 12. And then moisture. We usually remove and or bind moisture. Humectant substance used primarily in foods and cosmetic products to help retain moisture, sorbitol, and we also have dehydrant. And water activity AW, sebelum ni saya ada sebut AW, for measure of the amount of water within the substrate that an organism can use to support growth. Most fresh food, dia punya AW is more than 0.99. Okay. And microbial growth and AW, so halophilic AW dia more is around 0.75. Organism that thrive in environment with high concentration of salt. Halophilic, Xerophilic moles, organism that can grow and reproduce in condition with low availability of water. Osmophilic yeast, organism that are able to grow and in environment with a high sugar concentration. Lowest AW for pathogen growth, which is 0.86, which is Staphylococcus aureus. Ni gambar Staphylococcus aureus. So osmotic pressure, cells are 80 to 90% water. So dia memerlukan air. So hypotonic solution, high osmotic pressure remove water from cells, cause shrinkage of cell membrane, which is plasmolysis, used to control spoilage and microbial growth. For example, sugar in jelly and then salt in meat. So bila kita letak salt tu akan keluarkan air daripada sel. Okay, so akan kuranglah chances untuk bacteria grow in that food. Okay, hypotonic solution, low osmotic pressure causes water to enter the cells. In most cases, cell wall prevent excessive entry of water. Microbes may lyse or burst if cell wall is weak. Sebab bila kita letak sel dalam hypotonic solution, air akan masuk ke dalam sel dan akan menyebabkan sel tu pecah. So boleh tengok sini isotonic versus hypertonic solution of plasmosis plasmolysis, Plasmolysis refers to plasma membrane yang pecah, yang berubah. Okay, for example, kita letak sel, normal cell in isotonic solution, dia akan retain the plasma membrane. But when we put the cell in NaCl 10%, plasmolyzed cell in hypertonic solution, plasma membrane akan kecut sebab air tu akan keluar, will exit the cells. Okay, effect of osmosis in bacterial cell wall. Okay, contoh kat sini kita boleh tengok cell wall ada solute plasma membrane, ada water. So isotonic dia akan mengekalkan, dia akan, because there is no movement of water from inside or outside the cells. But in hypotonic, hypoosmotic solution, water will moves out of the cell and make cell to burst. Sebab maybe because of the cell wall weak of cell wall. And then hypertonic solution, water moves out from the cell. Sorry, yang hypotonic tadi air masuk and then akan menyebabkan cell to burst. But in hypertonic solution, air akan keluar so that plasma tu akan kecut. Okay, shrink of the plasma membrane, which is plasmolysis. Okay, so osmotic pressure, halophiles require moderate to large salt concentration. Ocean water contains 3.5% salt. Most bacteria in ocean. So extreme or obligate halophile require very high salt concentration, between 20% to 30%, and facultative halophiles do not require high salt concentration for growth, but tolerate 2% salts or more. Oxidation or reduction potential, the ease with which the substrate loses or gain electrons. So lost electrons kita panggil oxidize, gain electron kita panggil reduce. So we also have aerobic and anaerobic, microaerobic, and also facultative anaerobic. So you should know what is the term what is the meaning of the term here. Sebab dah belajar microbiology previously kan. So others, we have nutrients, fastidious versus non-fastidious bacteria that can produce their own vitamin. So they usually acquire them from the host. So they require special fortified agar to be grown in the laboratory. And then we have biological structure, antimicrobial factor, naturally occurring factors. Now we move on to extrinsic factor, which is temperature, relative humidity, gases in the environment, and also presence of other microorganisms. So temperature, we know that microorganisms grow over a wide range of temperature. Also, this include in the food microbiology, which is we have psychrophiles, mesophiles, thermophiles, psychoduric, and thermoduric. So, for example, kalau kita letak, if we put the food in the fridge, okay, what happened to that microbes? Do they become dormant, or do they stay alive, or what did they do? So basically, it depends on the type of microorganism. If that microorganism is a mesophile, what is mesophile? Dia suka temperature yang biasa between 4 to 27 basically. Sorry, 37. So dia akan dormant dalam peti ais. Fridge fridge. Sebab fridge is about 0 to 4. Okay, 0 to 4 degree Celsius. So dia akan dormant waktu bila kita bawa keluar daripada fridge, dia akan start grow balik. Okay. So temperature microbes are loosely classified into several groups based on their preferred temperature range. For example, psychrophile living can grow ranging between minus 15 to 10 degrees Celsius. And then we have two groups, true psychrophiles and psychotrophs. Okay, so sensitive to temperature over 20 degrees Celsius, optimum growth at 15 degrees Celsius or below, found in very cold environment, which is North Pole, ocean depth, seldom causes disease and food spoilage. Sebab kita tak operate makanan dekat North Pole ke, jarang-jarang orang duduk kat North Pole ni. And then psychotrop optimum temperature at 20 to 30 degrees Celsius, responsible for most low temperature for food spoilage. Okay, mesophiles, middling living, most bacteria usually mesophiles, okay, include most pathogen and common spoilage. Best growth between 25 to 40 degrees Celsius, optimum temperature commonly 37 degrees Celsius. So ni yang mana? Temperature badan kita kan. So many have adapted to live in the bodies of animals. So 37 is the optimum sebab dia boleh membiak dekat sini pada paling banyak. Okay. And thermophile, heat loving, optimum growth between 50 to 60 degrees Celsius. Many cannot grow below 45. So maksudnya kalau kita heat, bila kita continuously heat, macam contoh sup dekat apa nama ni, Pizza Hut, let's say, kan dia reheat. So maybe there are some thermophiles inside that because it is reheated around 50 to 60 degrees Celsius kan. So adapted to live in sunlit soil, compost piles and hot spring. Some thermophiles form extremely heat resistant endospores. Contoh ni lah, dia punya endospore kat sini. Sebab tu dia tahan the temperature. Okay, and then extreme thermophile, hyperthermophiles, optimum growth at 80 degrees Celsius or higher. Archaebacteria most live in volcanic and ocean vents. Contohnya lah, okay, you can see here different colours of surface dekat hot spring punya places ni, hot spring place here. So that colour is the differences in colours is the colour of the bacteria. Okay, here you can see yellow colour, orange your orange color here. So it is maybe different bacteria. Any green color, so different kind of bacteria. Okay, growth rate of bacterial growth at different temperature. So you can see here, psychophiles kat mana, psychotrops kat mana, mesophile mana, thermophiles and extreme thermophiles. Dia punya optimum here, for example, mesophile is 37, kan? And then thermophiles is about 61 or 62. And then extreme thermophiles is about 93, I think 93 or 94. So boleh tengok dia punya growth rate of bacteria. Okay, and then food spoilage temperature is about, kat sini, we usually call it danger zone, danger zone. Okay, because of rapid growth of bacteria, some may produce toxin here. And also, kita boleh tengok sini, this is degree centigrade, degree Celsius, and this is Fahrenheit. Okay, temperature at, in this range destroy most microbes, although lower temperature takes more time. Kat sini. Between 60 to 130 degrees Celsius. Between 50 to 60, very slow bacterial growth. And here is rapid bacterial growth. 0 to 15 degrees Celsius, many bacteria survive, some may grow. Okay, 0 to 4, refrigerator temperature may allow slow growth of spoilage bacteria, very few pathogens. Okay, between minus 30 to 0 degrees Celsius, no significant bacterial growth below freezing. Why? Why do you think it is no significant growth below freezing? So you should have the answer. Okay. Other factors which is relative humidity of environment, which is can change the AW, and also environmental gases, CO2 and ozone. And also requirement for growth, we have chemical requirement. The carbon makes up to 40% of dry weight of cell and structural backbone of all organic compound. Organism that obtain energy by the oxidation of electron donor and in their environment are called chemotrophs. Okay, heterotrophs obtain from their energy source, lipid process, and carbohydrates. Chemoautotrophs and photoautotrophs obtain carbon from carbon dioxide. So chemoautotroph, which is chemotrophic autotroph, chemical, auto self-drop nourishment. So they self-produce the nourishment. Okay, and then chemoheterotrophs. Chemical, hetero, other, trophs, nourishment. So dia ambil daripada luar. What about photoautotrophs? Can you find this? Photoautotrophs. Okay, nitrogen, sulfur, and phosphorus. Nitrogen makes up 14% of dry cell weight used to form amino acid, DNA, and RNA. Source of nitrogen, we have protein, ammonium, nitrogen gas, and also nitrates. And also sulfur, used to form protein and some vitamin, thiamine and biotin. Sources of sulfur, we have protein, hydrogen sulfide, sulfates, and phosphorus, used to form DNA, RNA, ATP, and phospholipid. Source mainly in organic phosphate, salts, and buffer. Other elements such as potassium, magnesium, and calcium are often required as enzyme cofactor. Calcium is required for cell wall synthesis in gram-positive bacteria, and also trace element. For example, many are used as enzyme cofactor. For example, commonly found in tap water, the iron, copper, molybdenum, and zinc. And also we have oxygen. Oxygen, organism that use molecular O2 produce more energy from nutrient than anaerobes, obligate aerobes. require oxygen to live. Disadvantages, oxygen dissolved poorly in water. Ni example dia Pseudomonas commonly nosocomial pathogen. And so we have facultative anaerobes. Example, E. coli, Staphylococcus, yeast, and many other intestinal bacteria. Obligate anaerobes cannot use oxygen and harm by the presence of toxic form of oxygen. Example, Clostridium bacteria that cause tetanus and botulism. Sorry. Botulism, Okay. We also have aero-tolerant anaerobes cannot use oxygen, but tolerate it present can break down toxic form of oxygen. For example, lactobacillus carries out fermentation regardless of oxygen presence. Microaerophiles require oxygen, but at low concentration, sensitive to toxic form of oxygen. For example, Campylobacter. So we have toxic form of oxygen. We have singlet oxygen, extremely reactive form of oxygen present in phagocytic cells. Superoxide free radical, O2 minus, extremely toxic and reactive form of oxygen. All organisms grow at atmospheric oxygen must produce an enzyme, superoxide dismutase, SOD, to get rid of them. SOD is made by aerobes, facultative anaerobes, and aerotolerant anaerobes, but not by anaerobes and microaerophiles. So kita boleh tengok kat sini apa dia buat. O2 minus tambah O2 plus 2H plus, ada enzim SOD, pun jadi H2O2 and O2. And then we have hydrogen peroxide, H2O2. Peroxide ion is a toxic and the active ingredients of several antimicrobials. There are two different enzymes that break down hydrogen peroxide. Catalase break hydrogen peroxide into water and O2, commonly produced by human as well as bacteria, and also peroxidase. Convert hydrogen peroxide into water. So that's all for today. And next lecture we will learn about microbial metabolism of food components. Metabolism of food components and nutrient in cytoplasm, cytoplasmic membrane, and also perisplasmic space in gram-negative bacteria. Okay, that's all for me. That's all from me. Thank you. If you have any problem with the notes or with the date, with the apa nama ni, with the apa nama ni, with the recorded lecture, please contact me. Okay. Have a nice day. Bye.